Alright, so we're going to pick up on our third installment of the metabolism chapter. Alright, so here you're being shown an image of 3 different types of metabolic pathways. Alright, so we've already talked about 2 of these forms. Alright, we've already talked about linear, we've already talked about branched. Alright, so for example, we can talk about a linear form. Alright, you have your reactant A, alright, that's converted into product B by some enzyme. Alright, for instance, enzyme 1. And then you have the product B that becomes the reactant of the second chemical reaction, and you form product C. Alright, and you have a second enzyme, totally different from the first, that catalyzes the second reaction here. Alright, now this is a long linear chain of reactions and comes to the formation of a final ultimate product. Now, the other type of metabolic pathway form will be a cyclical pathway. Alright, so for instance, they give the example of the Krebs cycle, right? Which we'll talk about in the next series of slides. Alright, but here you have some product from a previous pathway, alright, that's entering the cyclical Krebs cycle. Alright, so here you have the interaction of this reactant product form and this one, and it's used to generate this product here. Alright, this product is converted into X, X is converted into Y, Y is converted into Z. And so on. And this pathway replenishes itself. Alright, so you're always going to produce this molecule here. Alright, the pathway is always going to replenish what it starts with. And you have products that come out of the pathway. You have reactants that come in, you have products that pop off. Alright. Here you have a branch chain. Alright, now your branch chain can be either divergent or convergent. Alright, so if something diverges, alright, if you're trying to divert something, you're going in another direction, right? So here you have the initial reaction, you have M that's converted to N. Now N can go one of 2 ways. It can be converted into P or O. And this requires, 2 different enzymes. You can have enzyme 1 here, enzyme 2 here. Alright, 2 different enzymes. They catalyze one of the other steps. But in this case, this metabolic product can be diverted into 2 different pathways, 2 different paths. Alright, it's basically a crossroads for that product. And what exactly the cell needs. If the cell needs this product here, well, this will be diverted in this direction. If the cell needs this, alright, more, it'll be diverted in this direction. Alright, now, convergent, alright, so if 2 lines converge, alright, they're going to meet at a point. Alright, so here you actually have 2 separate pathways, and eventually their products merge to come together to form this final product. Alright, so it takes C, product C, plus product Z to make M. Alright, but product C and Z are a part of 2 separate linear pathways that meet together to form this. Alright, so this entire lecture is on cellular respiration and fermentation. Alright, so what exactly is cellular respiration all about? Alright, well, the whole purpose of cell respiration is to make ATP. And as I've already talked about, ATP is the currency, the money, that the cell uses to drive metabolic processes, right, chemical reactions, and to drive other cellular processes like movement. Alright, so for instance, movement of the flagellum requires energy. Alright, there's a cost. You also can talk about ATP being involved in transport. Right, so transport of substances against their concentration gradient, so active transport, or something like group translocation. These processes involve the use of ATP. Right, so basically you're taking biological molecules with potential energy stored in them, potential chemical energy, and you're basically extracting this chemical energy and using it to make a temporary high energy molecule called ATP. Alright, so cellular respiration is a redox reaction. Alright, so here you have glucose plus oxygen, and you end up with CO2, water, and energy. And you also have heat that's lost. So since this is a redox reaction, look here. Glucose, you have 6 carbons, 12 hydrogens, 6 oxygen atoms. What you're left with is CO2. Alright, so the hydrogen atoms in glucose have been donated to some other molecule. Alright, so glucose has lost hydrogen atoms. Alright, so in this process of losing hydrogen atoms, what happens? Well, you have the breaking of chemical bonds. You have the release of energy, and that energy is captured and transferred and used to make ATP. Notice you also have one molecule of glucose that has 6 carbons chained together. And at the end of the process, you end up with 6 CO2 molecules. Alright, so you've broken carbon-hydrogen bonds, and you've also broken carbon-carbon bonds. Alright, so in this case, glucose is oxidized to CO2, because in this case, glucose is losing electrons, so it's oxidized to CO2. Oxygen is going to pick up the electrons that are lost from glucose and a few hydrogen atoms, and it's going to be reduced to water. Alright, so notice here, this entire process is balanced. Alright, so your glucose molecule, you have 12 hydrogens. Alright, well, you have 12 hydrogens. Then here, you also have 12 hydrogens in your 6 molecules of water. You also end up with 6 oxygen atoms as well in your water molecule, which, when everything is tallied up, you end up with the same number of carbon atoms, hydrogen atoms, and oxygen atoms on the left as you do on the right. Alright, so this is kind of tying back into this idea of metabolic processes that take place in the cell as an assembly line. Alright, so here you have nutrients in the outside environment, alright, and they are brought into the cell, and now that they're brought into the cell's got to use them, right? It's got to extract energy, right? So you have catabolic reactions that take these substrate molecules that you brought into the cell, and what you do is you basically extract energy. And this energy, form of ATP and other precursor molecules, is used to synthesize new molecules. Alright, so you end up your building blocks. Alright, talking about your monosaccharides. Alright, talking about your amino acids. Alright, talking about your nucleotides. Alright, and also talking about taking your fatty acids and attaching them to glycerol. Alright, or even making your phospholipids would be another example of this. Alright, but here you take your building blocks, you have your polymerization reactions that chain and link the building blocks, the monomers, into their macromolecules. Alright, so your nucleic acids, your proteins, your lipids, and your carbohydrates. And all these things are used in a symbol to either, one, keep the current bacterial cell alive, to allow it to grow, develop, adapt, change with its environment, or it's used to make a new bacterial cell. Okay? Now, during this process of bringing nutrients in, right, the cell is going to use its catabolic processes to extract energy to make ATP. Now, one of the interesting things here is that ATP in its catabolic process it's very cyclical. All right, you'll see this a little bit later on, but to get the whole process of cell respiration going and moving forward, you actually have to invest ATP. All right, so here the cell is taking what it's bringing in, its nutrients, it's breaking that down, obtaining energy, using that to make ATP, and then using the ATP it makes to keep the entire process going again. Alright, so coming back to this idea of electron carriers. Alright, so recall that NAD plus and FAD are electron carriers. Alright, their whole job is they function as little bitty taxi cabs that shuttle electrons from one place in the cell to another. Now, specifically, it's going to be the cytoplasm to the cell about the cytoplasm to the cell membrane. Alright. So, for instance, here you have this biological molecule. All right, it's oxidized, and you basically cleave off two hydrogen atoms, basically electrons. Those hydrogen atoms, electrons, get transferred to NAD+. NAD+, when it accepts these electrons, it becomes reduced to NADH. Alright, your NADH molecule that's reduced will then show your electrons to the cell membrane. Those electrons get donated to proteins in the membrane that make up the electron transport chain. In the process of donating electrons, NADH is going to be oxidized to NAD+, and your initial protein in the electron transport chain will be reduced. Okay? Now, all this energy you captured that's been transferred to NADH and FADH2 is transferred to the electron transport chain. The electron transport chain is involved in the synthesis of ATP. All that potential chemical energy, stored energy, is then transferred to this chain to make ATP. And again, at the very end of the chain, right, you have your electrons. The two same electrons from here that were in here, all right, are going to be transferred to oxygen to form water. All right. So when you're talking about aerobic respiration, oxygen is the final electron acceptor. All right, because here the very last step involves transfer of the electron to oxygen, and you end up with water as a product. All right, so there are our three main catabolic processes we'll talk about. All right, the one you're most familiar with, or the ones you're probably the most familiar with, are aerobic respiration and fermentation. All right, so we're going to devote most of our time to aerobic respiration and fermentation. We will touch on briefly aerobic respiration. But if you look here and look at the total yield of ATP, aerobic respiration by far produces the most energy, the most ATP molecules. Anaerobic respiration comes in a second, and then fermentation comes in dead last. Alright, with only 2 ATPs produced. Alright, so since we're talking about bacterial cells, alright, it's important that you understand where these specific processes take place. Alright, so in terms of catabolic processes, alright, talking about glycolysis is going to occur inside of the cytoplasm. Alright. Talking about Krebs, cycle, that's also going to occur in the cytoplasm. Now, the bacterial plasma membrane, alright, this is where your electron transport chain and your ATP synthase enzyme is located. Alright, so here you're talking about the process of oxidative phosphorylation. So we're going to start with the process of glycolysis.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of two phosphates to the glucose molecule at the expense.
Alright, so here, talking about glycolysis. Alright, so looking at the word, glyco is referring to a sugar, and lysis is referring to actually breaking something apart, so splitting. Alright, so here you're taking a 6-carbon molecule of glucose and breaking it apart into two 3-carbon molecules called pyruvate. Alright, so this particular process occurs under both aerobic and anaerobic conditions. So basically that means in the presence of oxygen and in the absence of oxygen. Alright, so aerobic, presence of oxygen, anaerobic without oxygen, in this absence. Alright, so when you're looking at glycolysis, alright, you can divide this process into two main phases. Alright, an energy investment phase and an energy harvesting phase. Alright, so to begin with, alright, you start off with glucose. Alright, that's the initial reactant in the process. You have some ATP. You toss in a few NAD pluses, some ADP, and some inorganic phosphates. Alright, so here, the energy investment phase. Alright, so to get the ball rolling, alright, the cell has to invest energy into this pathway. Alright, so here you have glucose that's phosphorylated into glucose 6-phosphate. Alright, so if you recall, this idea of group translocation. Alright, so group translocation involves the movement of glucose into the bacterial cell, into its cytosol, through this process of group translocation, where when the glucose molecule enters, it's phosphorylated. Alright, so it's phosphorylated into fructose 6-phosphate. Alright, so that's one ATP molecule you've used. Alright. So now, that glucose 6-phosphate gets, alright, we talked about isomerization, converted into fructose 6-phosphate. Alright. And then you have another ATP molecule where you have phosphorylation. Alright, so fructose 6-phosphate is going to be phosphorylated again, and you end up with fructose 1,6-bis phosphate. Alright, so this bis phosphate is telling you there's two phosphates. Alright, which makes sense, because you've attached 2 phosphate groups to the original molecules. Alright, so this fructose 1,6-bisphosphate, it gets converted into this guy here. Alright, so you get 2 molecules of glyceraldehyde 3-phosphate. Alright, so now once you've produced this G3P molecule, 2 of them, alright, from 1 molecule of glucose, you're ready to enter the energy harvesting phase. Alright, so the energy harvesting phase, you have just this G3P molecule that enters, and you have electrons that are going to be transferred from G3P to your electron carrier molecule, NAD+. Alright, so NAD+ is initially oxidized, alright, and G3P has hydrogen atoms. These hydrogen atoms get transferred to NAD+, and it becomes NADH. And so NADH is now reduced. Alright, so now we have 1 NADH molecule. Alright, so next, what you're left with is 1,3-bisphosphoglycerate. Alright, so now the cell is going to take this molecule, and you're going to transfer a phosphate group from 1,3-bisphosphoglycerate to a molecule of ADP. And in this process, you produce a molecule of ATP. Alright, so this particular type of method of producing ATP, which we'll talk about in the next slide, but it's called substrate level phosphorylation. Alright, so because here your substrate is going to be your 1,3-bisphosphoglycerate, and you're going to transfer a phosphate group from that molecule to ADP, adenosine diphosphate, and you end up with ATP, adenosine triphosphate. Alright, so now we've made 1 molecule of NADH, and we've made 1 molecule of ATP. Alright, so later on down the line, alright, you have some more intermediate steps, and then you end up with what we call PEP. Alright, so this is phosphoenol pyruvate. Alright, so phosphoenol pyruvate. Again, phosphoenol pyruvate. Alright, so the phospho is a key thing here. Alright, so the phosphate from phosphoenol pyruvate gets transferred to ADP, giving you a molecule of ATP. And you're left with pyruvate. Alright, so here, when you're looking at the overall process, you end up with a total of 4 ATP. Alright, so basically, you take glucose, alright, you split it into 2 molecules of G3P. Alright, so total of 6 carbons. Alright, still. And then each of these guys enters this process here. And for each G3P molecule, you get 1 NADH and 2 ATP. Alright, so you get a total of 4 ATP. Alright, you get a total of 2 NADHs, and you get a total of 2 pyruvate molecules. Now, how many ATP molecules do you actually make? Wait, what is your actual net yield? Well, think about it. You had to invest 2 ATP molecules in the beginning. you made a total of 4. Alright, so the difference is really a net yield of 2 ATP. That's what you really made. Alright, so this kind of walks you through a more simplified model of the process of glycolysis. Alright, so you have your glucose converted into your fructose 1,6-bisphosphate, so this is where you've invested your 2 ATP. Here you have your G3P molecule here and here from the splitting. Alright, each of these G3P molecules is converted into a molecule of pyruvate, and the result is you make a total of 2 ATPs and 2 NADHs, and you end up with pyruvate, or pyruvic acid. Alright, so the end product of glycolysis is pyruvate. Right? Alright, and again, all this occurs in the cytosol of the cell. Alright, so looking at substrate level phosphorylation. Alright, so here you're given the example of phosphoenolpyruvate. Alright, so phosphoenolpyruvate, or PEP. Alright, this is the last step of glycolysis. Alright, this particular molecule binds to an enzyme, alright, called pyruvate kinase. Alright, so pyruvate kinase binds to PEP. You also have a binding site for ADP. Now, this enzyme brings together the PEP and ADP in the right configuration, the right spatial orientation with the right amount of energy, so that the phosphate group from PEP is transferred to ADP. Alright, so this phosphate group gets transferred to ADP, giving you pyruvate and ATP. Alright, so any enzyme that has the word kinase in it tells you that you are transferring a phosphate group from one molecule to another. Alright, so in this case, pyruvate kinase is telling you that you are removing a phosphate group from some molecule, in this case PEP, transferring it to ADP, and you end up with pyruvic acid. Alright, so this is telling you your end product, pyruvate. Kinase is telling you that there is some type of phosphorylation that's taking place. Alright, so here, this step, the production of acetyl-CoA. Now, this particular step, it depends on what textbook you pick up. Alright, some textbooks, alright, like to kind of lump together the production of acetyl-CoA and the Krebs cycle, and it's perfectly fine. Alright, but here, we're kind of teasing it out, separating it from one another. Alright, so here you have your pyruvate or pyruvic acid. Alright, notice that in blue, pyruvic acid, you have a carboxyl group. Right? So one of the initial steps involved in producing acetyl-CoA is that this carboxyl group has to be cleaved. And this is where you release carbon dioxide. Alright. One of the next steps is this, what you're left with once you remove this CO2 molecule, is an acetyl group. Alright, now this acetyl group is going to transfer electrons to NAD. Alright, so NAD is going to be reduced to NADH. Here you have NAD plus reduced to NADH. And also, this acetyl group is going to be attached to coenzyme A. So acetyl is attached to coenzyme A. So hence the word or name acetyl-CoA. Alright, so in this process, you actually yield 2 NADH molecules and 2 acetyl-CoA molecules. Alright, because the end of glycolysis, you produce 2 molecules of pyruvate. Alright, now you produce 2 molecules of pyruvate. Those 2 pyruvate molecules are converted to acetyl-CoA. Alright, so you get 2 acetyl-CoA molecules. Alright, in the process, each pyruvic acid molecule is decarboxylated and it's going to lose electrons, so it's oxidized, and NADH, or NAD plus, is reduced to NADH. Okay? Now, keep track of this. So far, we've had pyruvic acid. Alright, so pyruvic acid, so pyruvate. So pyruvate has how many carbons? 3, right? And you actually have 2 of these molecules. Alright, so since you have 2 of them, you actually have a total of 6 carbons. Now that's at the end of glycolysis. Alright, so now what we have done is we have cleaved off 2 CO2 molecules. Alright, so in the conversion of pyruvic acid to acetyl-CoA, you actually have removed 2 total carbon atoms. Alright, so now you're left with a total of 4 carbons that were originally part of glucose. Alright, so these 4 carbons were originally part of glucose. Alright, so you cleaved off 2 in CO2. So you're left with 4. Alright, so basically you gotta keep track of the carbon atoms. Okay? Alright, this process is also going to occur in the cytoplasm. Now, one of the key things here is that actually this step, as compared to glycolysis, this occurs only under aerobic conditions. Alright, so later on we'll talk about pyruvate, and we'll talk about how pyruvate, in the presence of oxygen, alright, it enters this pathway. Alright, so that's where we are. But when it doesn't, when there's no oxygen present, what happens to the pyruvate? We'll talk about fermentation later on. Alright, so here, your conversion of pyruvate to acetyl-CoA. Alright, this involves oxidative decarboxylation. Alright, so let's break that word down. First thing, decarboxylation. Well, D implies the removal of something, right? Carboxylation, your carboxyl group. Alright, so here you remove a carboxyl group, basically a molecule of CO2. Oxidative, you have a redox reaction. Alright, so acetyl is going to be oxidized into acetyl-CoA. and NAD plus is reduced to NADH. Now this process of oxidation, alright, so this process of transferring electrons from acetyl to NAD plus involves pyruvate dehydrogenase. Alright, so again, a dehydrogenase enzyme cleaves off hydrogen atoms, alright, which happens here. That little hydrogen atom goes there, and electron. Alright, so moving on to the Krebs cycle. Alright, so talking about the Krebs cycle. Alright, so the Krebs cycle is also referred to as the citric acid cycle. Alright, so here, this process again occurs in the cytoplasm of the cell. Alright, so here, alright, to start off with, you have your acetyl-CoA molecule, alright, that you have just produced. Alright, your acetyl-CoA molecule, the acetyl group gets transferred, right, so this little guy here, gets transferred to a molecule called oxaloacetate. Alright, now oxaloacetate is actually 4 carbons. Remember that the acetyl group is actually 2 carbons. So, now, you form this new product called citrate, or citric acid, that has a total of 6 carbons. Alright, so again, this is one of these cyclical biochemical pathways. Alright, so citrate is going to now go through a series of chemical reactions. Alright, one of these is going to be dehydrogenation reactions. Alright, so again, a dehydrogenation reaction involves the removal of hydrogen atoms from a molecule. They're transferred to other molecules, in this case, NAD plus and FAD. Alright, so this transfer of hydrogen atoms and electrons to NAD plus and FAD allows for this redox reaction to take place. Alright? So here, for example, isocitrate is converted into alpha ketoglutarate. It involves the transfer of the hydrogen atoms and electrons to NAD+. Alright, so in this case, isocitrate is oxidized to alpha-ketoglutarate. NAD+ is reduced to NADH. That same process occurs when alpha-ketoglutarate is oxidized to succinyl-CoA. Alright, and then succinate is oxidized to fumarate, but now the electrons are transferred to FAD, forming FADH2. Alright, next you have a decarboxylation reaction. Alright, so a decarboxylation reaction involves the removal of a carboxyl group, or basically removal of a molecule of CO2. Alright, so here what you see is isocitrate converted into alpha-ketoglutarate. You have a decarboxylation reaction that takes place. The conversion of alpha-ketoglutarate to succinyl-CoA, you have another decarboxylation reaction. Alright, so now you've actually removed 2 molecules of CO2. Now, this is important because at the end of the production of acetyl-CoA, alright, your single acetyl-CoA molecule is made up of 2 carbons. When you remove 2 carbon atoms, alright, you're basically taking that acetyl-CoA molecule, that acetyl group, and removing all the carbons from it as CO2 gas. Alright, so this pathway is referred to as a anaplerotic pathway because that's a fancy way of saying this pathway replenishes itself. Alright, so the citrate, molecule of citrate, is converted back into oxaloacetate. Now, let's track everything. Alright, so you start off with NAD+, FAD, ADP. You may start off with acetyl-CoA and oxaloacetate. Now, after one cycle, after one acetyl-CoA molecule enters the Krebs cycle, alright, you produce 2 molecules of CO2, here and here. You produce 3 molecules of NADH, and you produce 1 molecule of FADH2, as well as 1 molecule of ATP. Alright, this molecule of ATP is produced through, again, substrate level phosphorylation. Alright. Now, that's only one acetyl-CoA molecule. Remember that you had 2 molecules of pyruvate at the end of glycolysis. Each of those pyruvate molecules is converted to acetyl-CoA. So now that one is entered, the second acetyl-CoA molecule can enter. So at the end of Krebs cycle, the total amount of products that you end up with is 4 CO2 molecules, alright, 6 NADH, 2 FADH2s, and 2 ATP. Alright, so this is important. Alright, so basically, during this process of glycolysis, alright, tracking the carbons, alright, we've taken glucose, which is 6 carbons, alright, and we split it into 2 pyruvate molecules. Alright, that's 3 carbons, That's 3 carbons. Alright, so we haven't lost any carbons yet. So in the glycolysis, we still have the 6 original carbons in glucose. Alright, now, going from pyruvate to acetyl-CoA, alright, you lose a molecule of CO2. You lose a molecule of CO2. Alright, so now, if we tally it all up, alright, you have a total of 4 carbon atoms that were originally in glucose remaining. Okay, so now, let's look at what happens in the Krebs cycle. Alright, so now you have your acetyl-CoA molecule that is 2 carbons. It enters the Krebs cycle. Alright. In the process of basically going from citrate back into oxaloacetate. Alright. You actually remove 2 CO2 molecules. Alright, so 2 CO2 molecules means you have 2 carbon atoms. Alright, so 2 carbon atoms in acetyl-CoA, boom, gone. They're plucked off as CO2 gas. Alright, so the fact that this process happens 2 times means that you actually remove a total of 4 carbon atoms. Alright, basically 4 CO2 molecules. Alright, so you have completely, in this case, taken the molecule of C6H12O6, which gets oxidized to CO2, right? This is part of the reaction of cell respiration. Alright, so you've taken C6H12O6, or glucose, and you've oxidized to CO2. Alright, so in this case, you actually take, in the balanced reaction, you have 1 molecule of glucose with 6 carbons, and you produce a total of 6 CO2 molecules. Alright, it's the whole purpose of glycolysis and the production of acetyl-CoA and the production of, in the process of the Krebs cycle, is all about the complete oxidation of glucose to CO2. Alright, so we've only talked about the first half of the reaction of cell respiration. Alright, so we still have to talk about the second half, which takes place in the electron transport chain. Alright, so this is where we are. Now, ignore the mitochondrial matrix. Alright. Alright, so this is still inside of the cytoplasm. Alright. Ignore this little membrane here. Alright. We're not in the mitochondria. We're still in the cytosol. Alright, this is kind of walking you through the process. Alright, glucose is converted to pyruvate. Alright, you get 2 NADH, 2 pyruvate, 2 ATPs. Pyruvate is converted to acetyl-CoA, pluck off 2 CO2, transfer electrons to NADH. Now, you enter the citric acid cycle. You produce 6 NADH, 2 FADH2s, 4 CO2 molecules, and 2 ATP. Alright, so what is the whole purpose of this? What has taken place? Well, what you have done in this process of oxidizing glucose, you have removed electrons, potential energy from that molecule and the intermediates of that molecule, and transferred the energy to NADH and FADH2. Now, these electron carriers are going to shuttle electrons to the electron transport chain in the membrane of the bacterial cell. Alright, so when you're looking at the electron transport chain, alright, it's like water kind of moving downstream. Alright, so as you move downstream and you have incremental steps in the process, the amount of potential energy in terms of being transferred from one molecule to another decreases. Where the final step of this process involves transferring this energy, the electrons, to oxygen. Alright, so ignore the fact that this video is talking about the mitochondria, alright, because what's actually taking place is taking place in the cell membrane of the bacterial cell, alright. But here, the mitochondria is being used as the example. Alright, so here, scratch out mitochondrial matrix, membrane cristae. Alright, you do have an intermembrane space. Alright, but this is going to be your cytosol, alright, your bacterial cell, and this is going to be your cell membrane. Alright, now in this process you have your electron carrier molecule, alright, that transfers electrons to the protein complexes. Alright, so protein complexes don't consist of one protein. It's a protein complex. So there's many proteins interacting together that make up the complex as a whole. Alright, so NADH transfers electrons to this first complex. Right? Now, this complex is a component of the electron transport chain, which is a chain of intermembrane or membrane proteins. Alright, so NADH gives up its electron to the complex. Right? When it does this, NADH is going to be oxidized to NAD+, alright, and the protein complex is itself reduced. Now, this transfer of electrons provides the energy needed to pump protons against their concentration gradient out into this intermembrane space, this outside the cell membrane of the bacterial cell. Alright, so here you're actually building up a proton gradient, alright, where you have a high concentration of protons outside and a low concentration of protons inside. Alright. Alright, so now you have your next electron carrier, FADH2. FADH2 transfers its electrons to complex 2. Alright, FADH2, when it gives up its electron, it's oxidized to FAD. Complex 2 becomes reduced once it accepts these electrons. Alright, the electrons are then donated to these little intermediate carrier molecules, this guy here, that's going to shuttle the electrons to the next complex, alright, complex 3. Alright, so now, complex 3, when it accepts these electrons, it's going to be reduced. And these other two, in this intermediate molecule Q, is oxidized. Now this transfer of electrons to complex 3. Alright, this provides the energy needed to pump protons out into this intermembrane space. Alright, from the cytosol. Alright, next, your electrons are transferred to another intermediate molecule, cytochrome C, to complex 4. Alright, complex 4, when it accepts these electrons, it's reduced. You have the pumping of protons from the cytosol into the intermembrane space. And then finally, these electrons are donated to oxygen to form water. Alright, so here, alright, you have your C6H12O6 plus oxygen yields CO2 plus water plus ATP. Alright, well, we've talked about this already. Alright, that was glycolysis through Krebs cycle. Alright, that's where we oxidize glucose to CO2. Now, we're talking about taking oxygen that we breathe, and we're talking about reducing it to water. Alright, that's what's taking place here. Alright, the oxygen gas molecule accepts the electron from the cytochrome complex, and in doing so, it's reduced to water. Alright, so oxygen is the final electron acceptor because it's accepting these electrons in this pathway. It's the final electron acceptor. And the end product here is water. Alright, so we've talked about the oxidation of glucose to CO2. We've talked about the reduction of oxygen gas to water. Alright, so now we have only one product remaining to explain. We've got to talk about how ATP is generated. Alright, that's the last step of this. Alright, so again, disregard matrix. Alright, this is going to be the cytosol. Alright, so disregard matrix. Alright, cytosol. Alright, so what's taking place here is what you've been doing is you have transferred electrons to these protein complexes embedded in the membrane of the bacterial cell. Alright, so now when that occurs, you have the pumping of protons across this cell membrane into this intermembrane space. Alright, so you're building up a concentration gradient of protons outside, where they're higher, and fewer protons inside. Alright, so now what's going to take place is these protons are going to flow through a protein called ATP synthase. So these protons move through this proton pump, ATP synthase. And when they do this, alright, notice that here you have this little animation. Alright, which is basically demonstrating these protons, higher outside gradient, ATP synthase, protein. Alright, your protons flow through. And now when your protons flow through this pump, you have ADP and inorganic phosphates. Well, this provides the energy needed to take ADP plus inorganic phosphate and form ATP. Alright, so you're using the concentration gradient of protons that you generated through the electron transport chain to generate ATP. Alright, so this is what's referred to as an electrochemical gradient. Alright, you have a chemical gradient because you have a difference in concentration of protons outside and inside the, the cytosol versus the intermembrane space. Right? Now, it is an electrical gradient because since you have a difference in charge, alright, you end up with a charge on the outside and a charge on the inside of the cell. Alright, so here, this is giving you a little bit better picture of your protons. Again, scratch out mitochondrial matrix. Alright. This is your cytosol, because you're dealing with bacteria. Alright, these protons are pumped through the ATP synthase enzyme. Alright, so ATP synthase. Alright, this whole thing is ATP synthase. As these protons pump through, it provides this mechanical energy needed, this push, to allow for ADP and inorganic phosphate to form ATP. Alright, so this is kind of showing you an image of what it would look like in a bacterial cell. Alright, so here, this kind of gives you the bacterial cell picture. Alright, what we've discussed so far. Alright, so talking about this electrochemical gradient. Alright, so because you're pumping out all these protons out here, alright, you end up with a plus charge on the outside, and you end up with a negative charge on the inside. Alright, so you end up with a voltage difference, a membrane potential, a voltage. Alright, so you have an energized membrane, highly positive outside, negative inside. Alright, so as you produce ATP, this difference in voltage kind of dissipates a little. It's a less energized membrane. Alright, so a cell is constantly trying to keep the membrane energized. Alright, if this membrane potential is dissipated, alright, this is also referred to as a proton motive force. Alright, because think about it. These protons are pumping through this ATP synthase. It's a motor force. Alright, so it's causing this motor, this pump, to generate ATP. Alright, so a cell membrane needs to be energized. If this membrane potential is disrupted, alright, this could be very lethal to a bacterial cell if it's not corrected. If there is not a difference in the membrane potential on the outside and inside of the cell, that bacterial cell is dead. So you need an energized membrane to keep the cell going. Because the energized membrane allows the cell to produce ATP. Alright, so this ATP can be used to do a couple of things. So you've talked about active transport. We've talked about chemical reactions. All right, but also this ATP can be hydrolyzed to allow the flagellum to rotate. Alright, so in this case for motility. Alright, so let's look at a kind of a summary of where we are. Alright, so here, every NADH molecule results in the production of 3 ATP. Every FADH2 molecule results in the production of 2 ATP. Alright, so if you compare substrate level phosphorylation, going from glycolysis to oxidative phosphorylation, you end up with 4 ATP from just substrate level phosphorylation. Oxidative phosphorylation, alright, you end up with a total of 32 to 34. Alright, so 2 NADHs equates to 2 times 3, 4 to 6 ATP in glycolysis. Alright, FADH2, 1 FADH2 equates to 2 ATP, so 2 produced in Krebs, you get 4. Alright, so you tally all these up and get 32 to 34 ATP. You add that together, you get a total of 36 to 30 ATP from one molecule of glucose. Alright, now this is for aerobic respiration. Now, sometimes you have bacterial cells that use some other molecule other than oxygen as the final electron acceptor. Alright, so in this case you can use nitrate. Alright, so nitrate reductase, for instance, is going to result in a conversion of nitrate into nitrite, and you end up with water, which is your end product of glycolysis, as well as NAD+. Alright, they can go back to glycolysis and Krebs and pick up more electrons. Alright, so in this case, the final electron acceptor isn't oxygen, but nitrate. Alright, so this is an anaerobic respiration process. Alright, so here you're being shown this nitrate reductase. Alright, so nitrate is going to pick up the electrons that are shuttled between electron transport proteins, the electron transport chain, these membrane proteins. It picks up the electrons from this, and here these electrons are transferred, forming water, and you're left with nitrite. And you're also still in the process of this electron transport chain. You're still building up a higher concentration of protons outside than inside the cell. And you're still generating ATP through ATP synthase. It's just the nitrate is taking the place of oxygen. So nitrate is going to be reduced, you have nitrate reductase, where you end up with nitrite. Now, you can have other final electron acceptors, like sulfate, which can act as the final electron acceptor. Alright, so now we're going to talk about fermentation. Alright, so now we're dealing with an organism that, in this case, in the absence of oxygen, it's not using some other final electron acceptor like a nitrogen or sulfur compound. It's going to use fermentation to produce energy. Alright, so in this case, the electron transport chain is actually temporarily turned off, alright, for something like a facultative anaerobe. Alright, so here, in the absence of oxygen, pyruvate, alright, scratch that. You're not going to have pyruvate molecule converted to acetyl-CoA, or that acetyl-CoA enter the Krebs cycle, because you need oxygen present. Here you have your electron transport chain. Alright, so all this kind of gets temporarily turned off. In this case, pyruvate is diverted to a process of fermentation. Alright, so here the cell takes up an organic compound, like glucose. Alright, so in this case, glucose, you're going to take it, and in the process, you're going to take away some phosphate groups from it. Alright, so here you can have glucose, here, your glucose molecule, alright, it's going to be phosphorylated. You have your oxidized compound, alright, in this case, pyruvate. Alright, so conversion of glucose to pyruvate, you do make a few ATP, alright, but this is through substrate level phosphorylation. In the process of actually removing electrons from this molecule and transfer them to NADH. All right, so your organic compound glucose. All right, you do transfer some electrons in the process of making pyruvate. You do make some NADH. All right, you do make ATP. All right, so now you have pyruvate. Now pyruvate is going to be converted to some fermentation products. So something like, for instance, ethanol and CO2 gas. All right. Now this process of converting pyruvate, all right. So remember, you have no oxygen present. All right. So you got to take pyruvate, and it's got to go somewhere. And you've also built up this excess of NADH. But the job of NADH is to shuttle electrons to the electron transport chain. But in the absence of oxygen, the electron transport chain is temporarily turned off. Alright, and so your molecules of NAD plus NADH, they're finite. Okay? Alright, so that means that the cell only has so many that it uses at a time. So it needs to replenish the amount of NAD plus. Alright, so that means that NADH has to transfer electrons to pyruvate. In other words, as to, in this case, in the process, it's going to give up its electrons. You're going to have some intermediate molecule that's going to be reduced, and NADH is going to be oxidized to NAD+. So they can come back into glycolysis and pick up more electrons from glucose. Alright, so the whole purpose of fermentation is to recycle NADH back into NAD+. Alright, so we're going to mainly talk about two types of fermentation. Alcohol fermentation and lactic acid fermentation. Alright, so here you have alcohol fermentation to begin with. That's what we'll start with. You have glucose. Through glycolysis, you produce pyruvate. Now, this pyruvate is going to be then converted into acetaldehyde. Alright, so this is going to be the fermentation process. Alright, so pyruvate, you decarboxylate pyruvate first. You're left with acetaldehyde. Acetaldehyde is converted into ethanol. In the process of converting acetaldehyde to ethanol, you also oxidize NADH back into NAD+. Acetaldehyde is reduced to ethanol. Alright, so this is where you get your alcohol and you get your CO2 from fermentation. Alright, so taking a step back here. Alright, kind of correct myself on this image just a little bit. So kind of tying it back in with alcohol fermentation. Alright, so cell takes up glucose, alright, and through a process of glycolysis, it converts it into pyruvate. Alright, now that pyruvate gets converted into, in this case, acetaldehyde. Acetaldehyde is then converted into ethanol and CO2. Alright, so here looking at the second type of fermentation, lactic acid fermentation. Alright, so here, this is the fermentation pathway we're going to look at. Alright, so pyruvate is going to be reduced into lactate, and NADH is oxidized to NAD+. Alright, so what I like about these two figures is that it shows that the whole purpose of this fermentation process is to keep glycolysis going, because if you recall, glycolysis occurs in the presence and the absence of oxygen. Alright? So to keep the process going, guess what? You need NAD+. Well, if in the process of converting glucose to pyruvate, you make NADH. Alright, so the cell has to balance itself out because there's no oxygen, and so it has to recycle the NADH back into NAD+ to keep glycolysis going. Alright, because this is how it's making ATP. Alright, so this is kind of giving you kind of a different view of alcohol fermentation. Alright, so here you have your pyruvate, you decarboxylate it, so you remove this carboxyl group. You have your CO2, you're left with your acetaldehyde. Your acetaldehyde is then going to be reduced into ethyl alcohol. In this process, you transfer electrons and hydrogen atoms to acetaldehyde. NADH becomes oxidized to NAD. Alright, here, looking at lactic acid fermentation. Alright, so pyruvate is going to be reduced to lactic acid. NADH is oxidized to NAD. So it's here. Hydrogen atom there. Hydrogen atom goes there. Alright, so it gets reduced. Alright, so looking at products of fermentation. Alright, so pyruvic acid in bacteria and other organisms can lead to different fermentation products. Alright, so it's not always lactic acid or alcohol. Alright, so you can make cheeses, you can make wine, you can make beer. Alright, you can make you can make acetic acid or vinegar. Alright, so mixed acid fermentation. You can produce a mixture of products: acetic acid, succinic acid, alcohol, ethanol. You can produce propionic acid and acetic acid. You can also produce other gases. Right, so butyric acid, butanol, isopropanol. Right, so in this case, you're talking about your strict anaerobic or strict anaerobic bacteria. Sorry, they can lead to the development of gangrene. Alright, so kind of keeping a tally here. Alright, so comparing anaerobic to aerobic conditions. Alright, so anaerobic conditions, alright, in terms of fermentation. Alright, so anaerobic conditions specifically is talking about fermentation here. You make 4 ATP through substrate level phosphorylation. Alright, recall that to start glycolysis you need 2 ATP. So you gotta subtract 2, so you're left with 2 total. Aerobic conditions, aerobic cellular respiration. Alright, you produce a total of 38 ATP. 36 to 38. Alright, so this is kind of a nice overview of metabolism. Alright, so it's showing you aerobic respiration and fermentation. Alright, so here, all the electrons that are transferred to your electron carriers NADH and FADH2. All these electrons are transferred to the electron transport chain. And the whole function of the electron transport chain is so that you build up a proton gradient outside this intermembrane space so that the protons can flow back through the ATP synthase protein pump and make ATP. Alright, so the whole purpose of glycolysis in Krebs is to oxidize glucose. Alright, in the process of oxidizing glucose to CO2, you extract all that chemical potential energy, you shuttle it to the electron transport chain, where you generate a proton gradient, an electrochemical gradient of potential energy, used to generate ATP. As these protons go back down their concentration gradient through a process of facilitated diffusion. Alright, because your protons are going down their concentration gradient as they're used to make in the process, you allow for the production of ATP. Alright, so what is the maximum net yield of ATP per molecule of glucose for each of the following types of respiration? Alright, so remember here for these guys is about 36 to 38. Alright, you get about 36 or so ATP from anaerobic respiration, maybe less. And in fermentation, you get about 4 ATP. Or, sorry, 2 ATP. Alright, now, we've only focused on central carbon metabolism. Alright. Now, we've only also focused on just sugars. It's important to note that you have other types of biological molecules that can enter central carbon metabolism at different points. Alright, so for your sugars, they mainly enter initially as the initial step of glycolysis. Most. Alright. The other sugars get converted into glucose, and glucose enters glycolysis. Alright. Your fatty acids, remember they're made up of glycerol and fatty acids. So are your phospholipids. They have a phosphate group, a glycerol molecule, and fatty acids. Alright, so your glycerol, alright, so remember that glycerol can enter glycolysis. Alright, and it can be used to make pyruvate. Alright, your fatty acids can be used to make acetyl-CoA, right, the acetyl group. Alright, your amino acids from your proteins can enter the citric acid cycle or be used to make acetyl-CoA. Alright, so all this is involved in catabolism. Alright, because all these steps can lead to the production of energy, or ATP, as you break down your macromolecules into their individual monomers. You extract energy, and you use that energy to make ATP. Alright? The flip side of that is that you can take these smaller monomeric units and assemble them into larger macromolecules. Alright, so when something is in excess, alright, the cell can divert it to another pathway. Alright, so glucose can be used to make glycogen starch. Other intermediates can be used to make RNA and DNA. Alright, your fatty acids from acetyl-CoA can be assembled and then used to make your phospholipids and your fats. And then citric acid cycle, you can funnel off amino acids, or at least precursors of amino acids that you use to make proteins. Alright, so there's always catabolic and anabolic processes taking place. Alright, so you have synthetic and degradative processes. Alright, so this is why it's called a amphibolic pathway, or pathways. Alright, the ability of a cell to integrate molecules using and molecule building pathways to improve cell efficiency is amphibolism. Alright, so if you catabolize something, alright, you break it down. You anabolize something, or an anabolism, you build it. Metabolism is basically anabolism plus catabolism.